Thank you all. Thank you for being here. You should be happy to see me, because I'm the last person between you and the door. So, anyways, I'm here today to talk about connections, and a connection that we almost never get to see, right? And if you think about it, there's connections everywhere in your life. Be it your home, you see electrical wirings everywhere in your home, or plumbings everywhere in your home, or even laptops or cell phones. You have little circuits in these laptop electrical instruments. Or if you want to think a little globally, there's interstates or highways connecting cities, major cities, and even roads connecting homes, right? Or power grids, power lines, sending power to everyone, right? There's connections everywhere, and even there is what is called wireless. We still call it wireless connections. I know how hard it is to survive a few minutes without wireless, right? So I don't have to tell you the importance of connections. But I'm here today to talk about a connection which, like I said, we never get to see. If you think about all these other connections that I just told you, these are all designs or devices that we built. In other words, our brain built it, right? And it's funny because if you think about your brain, which is nothing but a huge mess of connections by itself, right? And if you want to understand your brain, we need to understand what kind of connections are happening in your brain. And in fact, if you look at your brain, like I said, your brain has got billions of specialized cells called neurons. And these neurons are special in such a way. They have a cell body, and they have projections coming out of them called axons. And what they like to do is they like to connect to other neurons within the brain, or other in a series of connections going to connect every other cells and tissues all over your body. And primary purpose of these connections is really to be able to control your movement, control any activity or function within your body, right? So how are these connections achieved? Right? And many scientists believe that our brain, which is nothing but a huge connections, are really hard to map them. In fact, there are several initiatives funded by several nations, several countries, to just map the human brain, which is called brain mapping, and that's being considered as the next big thing. And in fact, people are already thinking about ideas like, can we 3D print a human brain? If we know these connections, we might be able to 3D print a human brain, which might be able to essentially replace our brain, or even replace parts of the brain where it could be affected, right? So if we want to do all these things, we really need to understand the connections. We really need to know what sits there originally in the brain, right? And not only just understanding these connections, we also need to know what are these connections' purpose. What do these connections mean? What's the primary function of every single connections, right? While people are trying to understand these connections, what we are trying to do is we are trying to understand the function of these connections within the brain. And it is hard to understand these connections, leave alone our brain has got billions of neurons, which is really hard to understand these connections. What we do really well is we take our problems to other people. That's what we really do as humans, right? So, and that's what we do too. We are taking these problems to a simple organisms like fish. And several people are looking at several other organisms to do that. But what we do in my lab is we use a special kind of tiny freshwater fish called zebrafish. That's what we use. And these zebrafish can be easily handled in the lab, and they can easily breed, and we can breed them in the lab, and they can lay eggs. In fact, if you see these videos, here is a zebrafish tank. They're mating, and you'll see tiny little eggs falling through the tank to the bottom of the tanks. And the reason why do we need these eggs? Because one key aspect of our brain is when we are born, when we are growing as a baby, our embryos, majority of these connections within your brain are already laid out. So if you really want to understand how these connections are formed or what their function is, you really need to go back to these embryonic stages and look at these connections and be able to manipulate them. Right? So one way we ask how these connections or what these connections do is we go in, we manipulate these connections. When I say manipulate, we can disrupt these connections and ask, okay, what is really going wrong? So we can know the purpose of these connections. Right? So what we do is we collect these embryos, and this is what an embryo looks like, a zebrafish embryo. Right? And if you look here, up top, you're going to have a single cell embryo, meaning that's when they are born. This is what it looks like. You have single cell in there. And over a period of time, like 24 hours, you already have a nicely developed embryo, which has got already a brain that's developing. Like 24 hours, you already have a brain that's developing. Right? And in 5 days, if you look in here, in 5 days, this is a free-swimming larvae. Right? And it's self-sustainable, meaning by the time it doesn't have its yolk anymore, it has to find its own food. It has to escape from any kind of predation it might have. Right? And whatever food it needs to eat, it needs to be able to digest and excrete whatever it needs to do. So it is completely self-sustainable. And a lot of its nervous system is already up there. The brain is already built. All these circuits are born. They're controlling. So it's really easy for us to go in, take some of these organisms to ask questions like, if we disrupt some of these connections, what do they do? Right? In fact, what we do in my lab is we look at this fish to see these connections. And here is a zebrafish head, the larval head. And what you see is a head. But what I see is all these connections within the head. Here's the brain of the zebrafish, and you can see all these connections. In fact, you can turn the white light off, you can see these connections even more clear. And this is only a subset of connections within the brain. This is not all of them. Right? And this is not just within the brain. You could take the trunk, right? And it's the same thing. You could see a lot of connections going on here. Right? You could see there's plenty of connections. Every single tissue and muscle within your body are connected by this something that we never see, nerves and neurons. Right? So what are these functions? What do they do? Right? That's what we want to understand. And what we really do in my lab is we like to understand these functions, but we ask questions really related to humans. And one key question that we are asking in my lab is, really, can we create human disease conditions in fish? And the reason being, if we could create models, we might be able to understand what's really happening in humans. Right? And that's what we have tried to do here. We have tried to create an autistic model in fish. And the way we did it is we know in humans there are several genetic reasons for individuals to have autism. So we took these genetic reasons, we used those manipulations in fish, we went in, manipulated this fish, several genes, and we created fish which is autistic too. And I'm going to tell you about this one particular type of fish. Right? So here is, again, a neural connection within the zebrafish larvae. And you could see here some of what is called these neurons, which are in green. And you could see these connections that's coming out of these neurons called axons that's connecting to all over the muscles and really helping them in what's called locomotion. These fish are able to move, swim, because of all these what's called motor axons. It's kind of like analogous to what we have in our spinal cord. We have connections coming up, connecting to all our muscles too. Okay? And in the autistic fish we created, that's what you see here. You could see the difference here. Right? These are autistic fish that we created, and you could see how the number of neurons, you can see they're not as dense, one. Number two, you can see this connection. They are like disorganized. Right? And the other thing that you could see is they're really not reaching the target muscle. So two things that you would see is, one, there's missed connection. They're really not connecting to the targets. Second, there are a lot of misconnections because a lot of these are connecting here. You could see a lot of these are connecting here too. There's a lot of misconnections too. Right? So it's all good. There's a lot of misconnections. So what? Right? How does it really affect the individual organism? So what we did is we actually take this fish, and they otherwise look normal. That's the key thing. They otherwise look normal. We take this fish, we put it in these small trays, plates in them, and we actually put these plates into this machine, which actually records their movement. We could put them in these machines, which is like behavior analysis machines, and you could set the timer, and they will pretty much, there are high-speed cameras in there, they will record the movement of this fish over a period of time, however long you want to do it. And even you could set stimuluses like lights coming on and off, separate paradigms, and you could make it programmed so such a way the lights would come on being a stimulus, and you could see how the fish are reacting to these lights and things like that. Okay? In fact, that's what we did. And what you see here is a normal fish. I apologize for fuzzy video, but you could see here is the normal fish, and he's going to be moving slowly, and this is a real-time movement. And one of the things that you would see is he doesn't have any pattern. He's just slowly moving around, strolling around, happy-go-lucky. And he likes to explore. That's what they do, right? They like to explore. And while you're watching him, you could see the autistic fish too. And you could see a clear difference. You could see they are not able to move properly. They're not able to explore properly. And a lot of times they have what's called the spinning. They just go around this one particular thing, right? And actually, there are two different ones. And if you look at this one, it's not as bad, but he still, if you just follow him, he still goes around the circle. And he can have this normal movement here and there, but he would go on this circling movement every now and then, right? What's happening here? Right? What's really happening? We took some of these neurons, we caused autism to this fish, some of these connections are wrong, and these connections can really reflect on their actions and their behavior. Right? In fact, we can actually track every single of this movement. Right? If you look at these fish, we can actually track their movement throughout the period of time. And if you look at the normal fish, like I said, they like to explore. They really like to see their boundary, like what we do. We like to know our boundaries, right? We like to push other people's boundaries. That's what we do, right? So that's what they do. They like to know their boundaries. But if you look at the autistic fish, they really don't care. Maybe physically they're not possible to do it, but for a lot of reasons we think they are able to move here and there, but a lot of times they like to just stay in one place. And the other thing is, if you just look at them for a shorter period of time, you could really see they're circling around these particular regions. In fact, all of these can be quantified. We can quantify them in terms of how much distance they can move. We can quantify them in terms of whether they're rotating, clockwise, anti-clockwise. All these things can be quantified. I'm not going to talk about this today, but they all can be quantified. And what does this really tell us, right? What this really tells us, if you put it back into the scheme of taking neurons and these pathways, the connections within our brain as a highway, right? Just as in our normal life, right? We have problems all the time in roads. There's traffic all the time, getting clogged, right? Or the broken roads, right? Happens all the time. What do? How do we fix these things? We come up with ways, right? One, we can find collateral shunts or detour. We go detour to get to where you want to go. That's always we do. And the other thing we could do is we could always expand the road, right? Traffic, we could always expand the road. And in fact, could we do the same thing to this fish too? Could these neurons too? Wouldn't it be so easy if we could do that? In fact, that's what we are trying to do in our lab. We are really trying to see if we could find drugs that would actually expand or somehow make these neurons to connect, to grow new shunts, or to even expand, to make this membrane of these neurons expand, or in fact more permeable, to allow them to be able to send this neural activity more freely. And in fact, we've been doing this for a while now. What we do is we take this fish in these plates, and that's where zebrafish is, again, really neat, because you can take this fish in these plates, and you could put the drugs in these wells. And you could actually put it in this behavior software to see, okay, how do they behave? Are they even fixed now, right? In fact, we have been doing it for a while, and some of these molecules are not visible, which is good, I guess. These are proprietary material. That's good. I'm happy. But we've been working with our collaborator to find these new drugs. And one of the things that we have found recently is we have found some drugs which can actually make these fish move more than normal. Right? And not only make them move more than normal, they can even affect their rotation. They don't like to go on rounds anymore. So potentially we have hope. That's what I'm trying to say. We have hope. It's not all messed up. We have hope to even fix these things. Right? Lastly, I would like to end with one key thing. After all, what we see is connections everywhere in our life. Like, be it physical, right? All these traffics, all these highways, all these wired connections, be it physical, or be it completely metaphysical, like faith, right? Love, happiness, being sad, all of these. What we think now is pretty much is a function of connections that's happening within our brain, right? And I would like to say these connections, it's who we are. and our experiences are making us who we are, but our experiences can be translated into the connections that we have. Thank you for the opportunity.